that now. And I usually like to wait. It's coming right up on 3 o'clock now here, but I like to wait a couple minutes because people trickle in a little later sometimes. So,
yeah, Brian, why don't you just tell me when you want me to start, and I'll introduce Bruce.
great. I'm just gonna. I think I'm just gonna wait 2 minutes, kind of arbitrarily.
Okay, sounds good.
Love the earthworms across America tag.
Well, there's Rock Doc,
Can you hear me, Brian?
otherwise
Yes,
known
yeah.
as John Luchai.
Yeah, okay. You admitted me this time with my alias.
Well, I think anybody's welcome to join. So,
Even
the,
Luchai?
yes,
You'll have
even
to
Luchai.
edit the beginning of this out.
Well, I'll have to edit what I'll have to edit out. I'll have to alter your permission so you can't cause trouble for us.
Yeah, that's right. Yeah, definitely mute him.
So maybe 1 more minute here.
Right, sounds good.
Okay, I think we can go ahead and get started. I want to thank everybody for joining us for the final NAS seminar of the fall 2020 semester. It's been a semester, I will say that. I'm going to let Mike Draney introduce our speaker.
All right, thanks, Brian. Yeah, it's a real pleasure for me to be able to introduce Bruce Snyder to you all today. He is not only a UW Green Bay alumnus at the undergraduate and graduate level, but also one of really my star students for during my whole career. Bruce hails from the north side of Chicago, and I think he came to the UW Green Bay because of the reputation we had for environmental programs, and he was a biology, environmental science double major. While he was an undergraduate, he did a Coffin grant with me up in Toft Point, and also he started doing a summer research assistantship at the Field Museum of Natural History down in Chicago, and that's where he really became interested in millipedes, and he is now, an expert on the group, as I'm sure he'll say a little bit about. He stayed on for a master's degree in our ES&P program, and we looked at methods to sample millipedes, and then he went off, and I'm really proud of this. He went and got his PhD where I got my PhD at University of Georgia. So he was from the department, got a PhD in ecology, School of Ecology there. And then he went and became the director of student programs or student programs coordinator at the Kanza Prairie Long-Term Ecological Research Site. It's associated with Kansas State. And a few years ago, he became assistant professor in the Department of Biology and Environmental Sciences at Georgia College in beautiful Milledgeville, Georgia. And he's going to talk to us about soil ecology in a changing world. So take it away, Bruce.
All right, thanks, Mike, for that great introduction. Thanks, everybody, for coming today. And it really is an honor to be speaking virtually at my two-time alma mater. UW Green Bay holds a really special place in my heart, and certainly has framed a lot of the way that I approach the world. And so we will jump into this here. I thought it wouldn't be sort of going and giving a talk at Green Bay unless I could find some old pictures as well. And I took a look in the archives, and the only thing I could find were some pictures of Mike and I from that undergraduate research project at Toff Point. So here's young me and young Mike from about 20 years ago. And this really is where I got my introduction to doing research, and really sort of set me off on the path to the career that I'm on now. So today, as my seminar topic says, I'm going to talk about soil ecology in a changing world. And I'm aiming to talk about three things today. First, hopefully I don't need to convince anybody of this, but I'm going to make my best effort to anyway, that soil is really important, especially soil fauna and soil ecology. I'm going to talk about global change and hope to convince you that global change is really a multifaceted and complex thing. And then the third thing is to talk about the interactions between soil ecology and global change. And so I'm going to do this through a few vignettes of different research projects that I've been a part of, kind of at the interface between those two concepts. And hopefully throughout all of this, I can really draw out a lot of the complexities in both of these topics. I have a very large PowerPoint that's taking its sweet time and switching from slide to slide. So, of course, we need to get on the same page and talk a little bit about soils. So soils are very complex things, right? Do not call them dirt. Right? They are composed of not just solids, liquids, and gases, right, all the different phases of matter, but they're also living things, right? We don't yet have soil on other planets because we have not found living things on those other planets. Right? So beyond just being this structurally complex and physically complex thing, it also is a biologically complex thing. Soil function is super important as well. Right? Soils do a lot in terms of supporting ecological processes. Soils support lots of ecosystem services that we use, things like decomposition, nutrient cycling, filtration of water, not to mention all the more practical and applied uses for things like agriculture. And on top of that, soils also have a huge amount of biodiversity. And a lot of this biodiversity is really unknown. Right? We have not described probably the majority of species that are found in soils. And that sort of led to this science cover in 2004, calling soils the final frontier of biodiversity. Soils contain a gigantic range of taxa. So one of the great things about my current position is I get to teach a lot of focused upper level courses. We're a public liberal arts university. We're about the size of UW Green Bay. And I really get to have these nice small classes where I can focus in on things. And one thing I teach is soil ecology. And in that class, I cover at least 6 different phyla. And one of those is the arthropods. And within that, I cover all 4 of the extant arthropod subphyla. Right? And 5 bonus points to everybody who can name all of the arthropod subphyla. Okay, so we talk about all of these things on your screen living in soils, and a lot more as well. And we start to think about all the different interactions between all of these organisms, but also the abiotic environment, and you start to get a sense of the complexity with soil ecology. I was hoping to show a food web to kind of hint at that complexity. This picture is what the NRCS shows for a soil food web, and it's super simplified, meant for public consumption type of diagram. Even includes some things which are clearly not soil fauna. And I wanted to add in another figure, and this is a bit of an older food web diagram. And this is really just looking at, basically this section of this diagram. And in a fairly simple system, this is from a shortgrass steppe system, but it gives you a sense of just how complicated the system is and how many interconnections there are. And also consider that within each of these little boxes, there are lots and lots of species in any given soil system. Okay, so very complex, even in terms of ecological interactions. I'm only going to focus in on a couple of fauna. So most of what I do is, as Mike mentioned in his introduction, I have studied millipedes for quite a while now. One of the great things about when I came to University of Georgia is I worked with someone who specialized in earthworms, and he was thrilled to have somebody who would want to learn earthworms, and I was thrilled to be able to keep looking at millipedes. And so I have focused on these two big taxa. And so I will spend a little bit of time now talking about each of these groups in some fairly general terms. There are not a lot of earthworm or millipede specialists out there. So earthworms are really most well known as ecosystem engineers. So they physically change their environment, and that can really cascade to different effects on lots of other organisms. They do this through a lot of burrowing in the soil, mixing of the soil and also the soil in those leaf litter layers. They have pretty big effects on decomposition, especially those species that either eat the leaf litter or drag the leaf litter underground. And that also in turn has big effects on nutrient cycling. They also can affect things like aeration of the soil and filtration of water, or the ability for water to infiltrate the soil. Earthworms' ecological effects have been known for a very long time. Darwin wrote a whole book on earthworms, right? The formation of vegetable mold through the action of worms. And this even kind of got included into some of the Darwin got made fun of a lot because of his new views. And so this cartoon on the screen is from Punch's Almanac, when they're kind of poking fun at Darwin, saying man is but a worm, and trying to show the connections between earthworms and humans. So we've known about earthworms and their important activities for a very, very long time. And they still remain of interest to even the general public today. Most people know about earthworms as bait for fishing. Sometimes they pop up in other sorts of general interest things. But even though people are pretty familiar with earthworms, they don't really understand earthworm diversity. So a lot of you probably recognize this earthworm, Lumbricus terrestris. I'm sure it's present in Wisconsin. It's present in most of the U.S. It's a European earthworm. This is the night crawler. But most people think that this is the earthworm, as if there was one species of earthworm. In reality, earthworms are a pretty diverse group on their own. There are about 3,500 species of earthworms currently described in 16 families. They have a really much bigger than you would think range of sort of size and color and lifestyle. So things from this native small called the American logworm, it's a few centimeters in length, to some much bigger earthworms that can be meters long. So this picture on the right here is from a colleague of mine down in Brazil, and they were down there and they sent an email and said, we're digging up earthworms with a backhoe. I said, I don't believe you. Send pictures or it didn't happen. And so they sent a picture of a backhoe and this picture, and it's hard to read, but on this measuring tape, that's 5 feet. So these are incredibly big earthworms. So pretty important in those systems. And they can also be colorful. So here is a species described out of the Philippines, which they sometimes call the fried egg earthworm. Very, very colorful. Earthworms are also globally distributed. Essentially, everywhere. So every continent except Antarctica. In North America, there are only about 170 species, and the distribution is shown here on the right in the gray areas. Earthworm distribution in North America is really closely tied to the Wisconsin glaciation, which is something that I'm guessing most of you are familiar with. At that time, 15, 20,000 years ago, they were really pushed south of that line. And you can see from the current distribution, they really have not come much further up above that line. They're fairly restricted to a group in the southeastern US, which sort of pushes towards the middle of the continent, and then there's a separate fauna on the west coast. There's also about 30% of these 170 species are non-native. Okay, primarily they are European and Asian species, and their distributions don't match this pattern. They are very widely distributed, and we'll take a little bit of a look at that later. But we don't have any big, bright, colorful, exciting species here. We have ones that look like the ones you see on the screen now. They do get fairly large. This Diplocardia species, it's very common down here, can get to be 12 to 14 inches or so in length. So the other group I work on are the millipedes. Millipedes, similarly, are also mainly detritivores. They're really known more for doing fragmentation of litter, chewing up that litter, breaking it into smaller pieces, getting the microbes moving on that. Their big claim to fame is that they were one of the first land animals. So our oldest fossil of a land animal is of a millipede. And this picture is of a model from, I believe, the Field Museum of one of these early millipedes, and these are about 2 meters in length. Okay, so they were much bigger than they are now. Okay. Millipedes are also very, very diverse. I'm going to try and stay out of the taxonomy too much today, but just to give you a sense of how diverse they are, there are about 16 orders of millipedes. Insects are in the low 20s, depending on who you ask, of how many orders there are. There are about 15,000 species that are currently valid, and of those species, about 1,500 are in North America. Millipedes are also incredibly diverse, right? Across 16 orders, you're bound to get a lot of different sizes, shapes, color patterns. They've got lots of really neat defensive adaptations. There's some that are bristly to fight off ants. They have lots of different chemical defenses. Some of them can coil up entirely, like in this picture in the lower right. Right, that's a pill millipede and is not an isopod, but a lot of them just coil up as well. And so yes, everything on this screen is a millipede. So you can probably see why I kind of fell in love with this group when I started to research them and really start to read about all their neat adaptations. So I'll shift gears a little bit and talk about our changing world. And when I say changing world, or when I talk about global change, what comes to mind? Well, for some of you, it may be the upcoming Georgia Senate runoff election. I know that's been in the news a lot. I dropped off my absentee ballot yesterday. For a lot of you, it's probably this. Now, people thinking mostly about global warming, global climate change in particular. But global change has really kind of more of an umbrella term to incorporate a bunch of different driving forces that we're seeing having really widespread impacts, right? Truly global impacts. Of course, climate is a big part of that, And it's more than just temperature, right? We're seeing lots of changes in patterns of precipitation and the timing of that precipitation as well. And as an ecologist, when I'm looking at this, especially at a more local scale, thinking about what earthworms and millipedes are experiencing, there are a lot of other big drivers. So pollution is something that is definitely happening at the global scale. And I'm not just thinking about PCBs or heavy metals, but also things like light pollution, which is potentially affecting soil fauna that are used to being in the dark. Sound pollution, because a lot of these species can feel vibrations. I've been working on nitrogen deposition, because that's a big issue, especially in the southeastern US, but is also a global phenomenon. We'll probably not have time to talk about that today, but be happy to answer any questions on that later. We also have land use and land cover change, and that can have some major impacts, especially at the landscape scale, connecting those populations. And also invasive species, which again is a very big global problem. And to be honest, the first time I put this slide together, I thought, you know, I really hate this figure. It was fine in PowerPoint, but it's an overly simplistic view of these problems. Right? They are not necessarily parallel. There are a lot more drivers here. There are a lot of connections between these different drivers that's all impacting the situation, and you really can't show that with a couple of arrows. In addition, these are each going to be affecting that complex soil ecological web in a number of different ways, and you're going to get outcomes at different scales. And so I want to show you a couple of things that I think will help to sort of set up more of a framework for thinking about how these drivers can potentially be affecting soil ecology. So I had a chance a couple of years ago to work on a little bit of the climate side of this. So with my good colleague Matt Callahan, who is at the Forest Service Research Wing and the Southern Research Station, just up the road from me in Athens, we got invited to write a chapter for this book, Ecosystem Consequences of Soil Warming. And they were taking a really broad view, but focused specifically on soil warming and how the warming of soil affects everything, essentially. So they asked us to look at the fauna, and we thought, hey, there's probably not that much research on this. This would be relatively easy to put together. And one of the challenges we found was that nobody looked at the same thing. There were all kinds of different responses they looked at, and all kinds of different ways that this happened. And so we ended up shifting and making, this is the reason the word potential is in our title. All right. We didn't talk much about the responses. We talked about, okay, what's a framework we can look at for trying to understand the way soil fauna may respond to warming. And I want to show you two figures out of this paper. This first one is, one of the reasons that we took this approach of setting up a framework is that soil is complex. And so not only do we have the interactions between the different parts of soil, and not just between the organisms, but also between the organic matter and the vegetation inputs. But we also have both direct and indirect effects. And so not only does temperature have some direct effects that can carry through, but temperature and moisture are inextricably linked, especially within soil systems. I think we know generally this is true in ecology, but it's a very tight linkage in soils. And so through moisture, you have a lot of indirect interactions that can then also change the responses that different parts of the system are going to have. And over on the right side here, we also have disturbance. One of the big challenges with working in soil systems is to get into that system. Because of its nature, you have to disturb the system. And so you can have that also causing some indirect effects and ultimately changing the response that you see. The other thing we realized was that this is happening across scales. And so we're thinking about global climate change and global warming in specific. We're thinking about 50, 100 years or more because that's what the models have been trying to predict. But when you're doing these experiments, you're not doing a 50-year experiment. You're doing a relatively short-term experiment and changing things more on the order of days to weeks to months. Okay? The responses also are happening at those different scales. Right? So you may turn up the temperature on your millipedes, and then they're going to have short-term responses to that. In addition, these are happening across levels of the biological hierarchy. Right? So you may be measuring individual behavior changes, or you may be measuring changes in community structure, and those are going to happen at different scales. And so we put together this figure to try and incorporate that and to show where you may actually have some overlap or some connection between them. Because if you do a research project and you look at behavior, and it's going to be an individual level trait, you're going to see changes over relatively short time scales. That change doesn't necessarily carry over to something like even community structure, right, which is going to be happening at a much longer time scale and definitely at a different scale in the biological hierarchy. And when we start thinking about the things that we really are most concerned about, extinction, novel ecosystems, then there's a bit of a disconnect between the types of experiments we're doing and the types of responses we're trying to look at. And so hopefully between these two things, you're getting a sense of how complicated the picture is, and probably also that we have a long ways to go. Right? We don't have research that covers this entire chart. And so I want to now move into some of the different things that I've tried to do to address some of these questions that we have about the interaction between soil ecology and between climate change. So one thing that I think is really critical if you want to look at how is global change going to change the soil fauna is to have a baseline to start with. And one of our big challenges within ecology, and especially within soils, is that we don't have a good baseline. Right? There are not a lot of soil ecologists out there, and for some of these groups, there are relatively few people that work on this group. Right? And so this is very true for earthworms and millipedes. And here I have a look at earthworm, or basically our more or less current state of knowledge as far as earthworm distributions go. So what's shown here is county scale data, and this is specifically data for Lumbricus rubellus, which is one of our European species that's pictured here on the right. These the black counties are ones that have Lumbricus rubellus present. The gray counties are counties where we have data for any earthworm species. And the white counties are ones where we do not have an earthworm record. Now this is based mostly on literature. There's mainly one guy who has gone around and tried to do checklists for every state. And there's a little bit of museum records in here as well. I'm sure there's a lot more in museums that we could add to this. And this data is a couple years old, but the main picture hasn't changed very much. And that is that we have a lot of work to do. Our data is very, very incomplete in terms of just knowing that which earthworms are present at the county scale. And some of this is a little bit of bias. Right? So Kentucky is very easy to pick out. Clearly the Kentucky checklist either has not been published or at least was not published at that time. And that's true for a number of states on here. But also when you look at a state like Georgia, there's a lot of open spaces. And whether that's just he didn't dig holes in those counties, or he dug holes and didn't find anything, is not super clear. So there's a lot of work to go on this. One approach that I've tried to take to address this is a community science approach. Or citizen science is another term for that. We're starting to shift and call it community science, which I think is a fantastic way, a much better descriptor of that approach. And so while I was teaching at Kansas State University, I got a little bit of money to do a project we called Earthworms Across Kansas. And our goal was, okay, we don't know what's here. There's a little bit of data, as you saw. Let me jump back to that previous slide. Here's Kansas, right in the middle of the country. It's this big open square, and not a lot of records. I thought, what would be a great way to do this, but to go ahead and try and actually get the community to help us collect things, because I cannot go to every county in Kansas, and it would be fun to do, but I've got a teaching load. I don't have the time to do that. And so what we did is we made these little kits, and actually, the great thing about being in your office is you can have props, and you don't have to carry them to the site. And so we had these little kits. The contents are all there on your screen, and we sent these out to school teachers. We recruited teachers all over the state. We asked them to go out, take your class out, collect anywhere. It's all going to be new data for us. Teach them something about soils or earthworms or ecology, whatever fits best in your schedule, and send us the specimens. And so these had a little prepaid sticker on them, and they would send the kit back to us with the specimens in them. And we would go through and process and do the IDs, because the IDs are not the easiest. And we had really great response and got a lot of data. And so these are where all the collections came from. We had this up on a website at the time, which I think the website's defunct now, but we had 300 people sign up, got over 100 kits back, 3,000 specimens, and actually got a lot of new data. So at least 5 species new to the state, and this is where you run into sort of the taxonomic challenges as well. So even with the experts in the country looking at these specimens, we have 3-ish new to North America, including a family that had not been, it was known but has not been published yet. And so this seems to be a pretty effective approach at getting at some of this problem. But I would say we still need to have more, get more people who can identify earthworms. In terms of millipedes, I really wish I could put up a county map just like the earthworm one, but nobody has ever pulled that data together. And with the diversity being so much higher, an order of magnitude higher than it is for earthworms, it's a big challenge. So I've been trying to do this in my position here at Georgia College. Georgia has the benefit of being very warm and very wet, which is fantastic for soil fauna. We have 10 of the 16 orders of millipedes here, and we've been working on a checklist draft. There we go. And so I had an undergraduate who was here, started to put together a draft, and he's actually coming back in the spring as a graduate student, which is fantastic. And he's going to continue to work on this, just using published records and my own specimens. Again, we have not gone back to the museums to find everything. We've got about 78 species, but based on this map, less than or about half of the counties have no published millipede records. And clearly it's not that there are no millipedes in those counties. Looked for them, right? Nobody's gone and collected them and actually brought them back. So we're expecting to add to this a lot. We know there are some undescribed species, which is really exciting to work in a group where you can go in your backyard and probably find an undescribed species. We know there are a lot of really common non-native species of millipedes that are probably, in every house across the state. So those are not incorporated here. And we know, as I said, that the museum specimens are there's a lot there that we can dig into. One of the really interesting things about this data, and this is a bit of an aside, but I think it's interesting, there's this giant hole in the distribution where it's not near any major interstate. There are no major cities here. You know, the people who have studied millipedes have clearly gone down the coast and they've gone to the big places, but there's nothing here. I am here in the middle of the state, which we do have records there now, but it's not filled in on the map yet. But it's really interesting. You start looking at all the distributions, and here are some maps from a selection of different millipede groups, and you see this pattern on every single map. And so we have some work to do, where we have this sort of black hole in the middle of, or in the south end of the state, and just nobody's been looking. So that is sort of in my list of things to try and tackle over the next few years. So I've already mentioned non-native species in both millipedes and earthworms, and invasive species are one of these big drivers of global change. And so I do want to spend some time talking about invasive species. And I'll skim through this pretty quickly because I think a lot of the audience knows what invasive species are. And I'm just going with the definition at the top there. Thinking about non-native species, which are causing some sort of damage. You're familiar with a lot of these different examples of that are really very obvious to most people. And we could spend a whole nother lecture just listing off other examples. But a lot of people forget or don't know that invasions are happening below ground as well. And so if you think about the earthworm or really the soil fauna in Green Bay or other urban areas, most of those species are at least non-native, if not invasive. And I know for a fact that these two pictures in the bottom right were taken in Green Bay. So these are, in fact, these are both European species, and they're very common throughout of developed North America. Earthworms in particular have been a big issue. It's been in the news a lot lately, which we'll get to in a moment, and it is a global problem. This is a figure out of a review paper we did in 2008, and each of these numbers represents a family. The take-home message here is that in each of these that are in a red or orange circle, there are individuals from that family that are invasive in that biogeographical province. And this is something that's true around the globe. Australia's got it really bad. But every single one of these regions has invasive earthworms. So it truly is a global issue. And usually these are from multiple families coming from multiple different places in the world. Earthworms have been introduced in lots of different ways. A lot of it's been unintentional. Soil used to be used as ship ballast, so certainly some things came in that way. We used to bring in soil with plants. Soil is still moved around with plants quite a bit. And again, people knew that earthworms were good for the soil. That's the way they thought about them. And so they were brought in intentionally for agricultural use. They've been used as bait and are still used as bait. Almost every bait earthworm is non-native. And these continue. Anytime there's earth-moving activities, there's the potential for earthworms and other soil fauna to be moved around. So whether that's construction or road building, the bait movement, of course, and horticulture as well. So from nurseries outward, probably. This is a photo from my colleague Mac, who bought a blueberry plant at one of the local stores and took the plant out, and in the bottom there were 4 earthworms. So this is happening, and we don't really have a great documentation for that, for how, in ecology we like to quantify. We don't have good numbers on how often this happens and how many species are being moved. Okay. This is another picture that I think shows some of the movement that's probably happening. So this is the picture of the gutter and the curb outside of my house in Kansas, and I was clearing out this grass, and probably hard to see on your screen, but there are a couple of invasive earthworms right there. And so these are moving around and being transported continually. Some of these earthworms can cause some really big ecosystem-wide changes. Okay? The most obvious impacts have happened in basically places where you are now, in those northern forests in Minnesota, Wisconsin, Michigan. There have been a number of studies in New York and in Alberta as well. Right? And these are places above that glacial maximum where there are no native earthworms. And so adding a new taxon to this system, one that a taxon that's so different, really can change a lot of things. So you can go from an understory that looks like this, got some nice sugar maple seedlings there, and it can become something like this. And this is mainly happening because of a European species in the family Lambricidae. They come in, they eat all of that nice leaf litter, and they're doing a lot of mixing of it as well. And that changes the forest floor. Right? It's going to lead to a lot of erosion and subsidence potentially, which is what you see in this bottom picture. And you can imagine if that's changing that physically so much, it's going to change nutrient cycles, it's going to change the microbes and really all of the fauna. Plants can't regenerate in this, especially these tree seedlings. Rare plants can be affected, and so it could be a major, major problem. So most of the research until about 10 or 15 years ago was focused on these European species. When I started working on earthworms, I was introduced to the pheredamoid earthworms. They're in the genus Ementhus and Pheredima and a few other genera, which I usually just call Ementhus and friends. These are Asian species, and we now have 16 species in the US, and here's pictures of a few of them, which I know to most people just looks like a bunch of worms. Right? These all look very, very similar. They're challenging to ID, and this is a picture out of a key that we wrote a few years ago, which is very helpful to this process. We have 16 here. There are hundreds of these species in this group in Asia. This group has really gotten a lot of attention recently as sort of a second wave of earthworm invasions. They've been in North America since the 1800s, but we're starting to find them more and more. There have been some newly discovered invasions in Wisconsin. Down at the UW Arboretum, where actually a classmate from my master's group, Brad Herrick, is leading up some of these investigations. There's been more reports in the Northeast. Maybe they're expanding, maybe not. And my contribution to all of this was this paper at the bottom, where we actually found a species that had not been seen in the Western Hemisphere before. One of my REU students in Kansas found this. And again, I got some props. These are not, here's my hand for scale. These are not small earthworms, right? So it's not like we're missing tiny, tiny things. These are very large earthworms that are around, and we're just now finding them. And so basically every time we stick a shovel in the ground, we're finding either species new to science or new invasions. So I want to talk a little bit about my research on one of these Ementhus species, Ementhus agrestis, and they were invading into the Smokies. They've, as I said, been known since at least the 1880s, and they've been in the Appalachian Mountains at least since the early 1970s. We didn't know they were moving into the park until 2003, and I started at University of Georgia in 2004 and began studying this invasion. These earthworms, unlike a lot of the other invaders, are living up in the leaf litter and mainly consuming leaf litter. And the agrestis name is pretty accurate. They're fairly aggressive. You pick them up, they will thrash around, they will self-amputate their tail. As you can see in this picture here, that tail is actually separated, which is a little bit freaky the first time that it happens to you. But beyond that, their biology and their ecology are very poorly known. Okay. So we've tried to tackle this in my dissertation work, and since then, through a combination of field and lab studies, to try and really understand what are the impacts and how are those changing. And so we know that they greatly reduce the amount of leaf litter. So in my field study in the Smokies, this is what a core out of the plots looked like. Had a nice amount of fresh leaf litter and then a huge amount of partially decomposed leaf litter. And when Amenthus agrestis comes in, they greatly reduce mostly that partially decomposed leaf litter. Okay. Unlike a lot of places in the north where an earthworm invasion comes in and you can see the change, you couldn't really see this because that top layer was still there. But when you really got in and started looking, you'd see that that partially decomposed litter was gone, and there was a layer of these sort of granular casts. And I have some in the lab, and so I took another picture yesterday of what the soil looks like in this container in the lab. So these are really big chunks of soil that they are creating, and that changes, again, the habitat for things like millipedes. So we also found in this field study that when the earthworms moved in, there were fewer millipede species, a lower abundance of millipedes as well. And millipedes in the Smokies are some of the most obvious, big, colorful invertebrate fauna that are there. And so this was a pretty big impact that we found. And that's what these graphs on the right are showing. Across the x-axis, we basically just have because of the way that this project went, I had to have kind of an index, so more frequently invaded to the right. And then we have millipede density in that bottom graph and species richness in that top graph. Pretty significant declines when you had those earthworms present. And it makes a lot of sense because earthworms and millipedes are, at least these earthworms, are living in the same part of the system. They're probably eating the same things, and certainly there's a lot of conversion of that leaf litter to a habitat that's really just not great for most millipedes. So I want to, if I've got the time, go through one more little vignette of some research that we're doing right now. And this is taking a look at the responses of soil fauna to wildfires. So we know with global change, especially with the changes in climate, we're having increased temperatures. We know that there is a predicted increase in droughts and severe droughts. And when you combine those two things, you set up the perfect situation for an increase in frequency and intensity of wildfires. I know there's been a ton of discussion about that with the California fires recently, and before that it was the Australia fires, and before that it was the Amazon that was on fire. So this has been a topic that's been out in the news a lot. My very first fall here, we had a pretty decent drought where for a couple of months we did not have rain, and that's very strange for down here, although it's happening more frequently. Because of that, we had a lot of very large and very intense wildfires in the region. Now, that's not that strange to have a lot of fires, right? Basically all of the Southeast is a fire-dominated system. But you also have to keep in mind that over time, yes, we have more, some years with more fires, some years with fewer, but in the early part of last century, we suppressed fire. And so while we've brought back fire to a lot of places in the Southeast, there are also a lot of places that have not burned in decades or more. And so when we have these droughts now, if fires get into places that have not burned in a long time, there's a pretty big fuel load. And that becomes very problematic because you can get much more intense fires than you would normally see. We're working in areas, again, with really high millipede diversity. So I'll show you in a minute. This research was done in northern Georgia, which is fairly mountainous, and up in the Smokies as well. And to layer on top of that, these are all sites that were invaded by these Amenthus earthworms. So we're able to take a look at not just what is the effect of fire on the millipede communities and the native earthworm communities, but also, is fire potentially going to help us with these earthworm invasions? You know, is it a way that we can control those invasions? And so we were funded by the U.S. Forest Service to do this work. Again, it's a collaboration between myself and Matt Callahan up at the Southern Research Station. We were able to find fire lines where we've got burned on one side of the line and unburned on the other side of the line. So we're only 10, 20 meters apart from each other, but they've had some very different impacts by the fire. And so we were able to start this right away in 2017, and we've been trying to collect seasonally since. Of course, the pandemic has thrown a big wrench in all of that. We haven't sampled this year, but we'll see what happens next year. Here are our sites. Again, we've got 2 in North Georgia, sort of northeast and northwest Georgia. Back a second here. And here's a picture of the Chimney Tops 2 fire, which is the one in the north side of the Smokies. This one burned into Gatlinburg and was in the news at the time. A lot of people lost houses. A few people lost their lives because of this fire. So we were able to kind of treat this as a natural experiment. Use these fires that were already happening. We didn't have to light any fires, and we could look at the effects on the soil fauna over time and see what the response was. It's really fantastic to have those resources nearby. Gatlinburg's a 5-hour drive, which is not something you can do overnight, but it's convenient enough that we can go for a weekend with students and get them involved, and I really enjoy that. So we took a look at the leaf litter dwelling fauna, so primarily the millipedes and also those Amenthus earthworms. We did a timed hand collection within a 2-meter radius, which of course makes it challenging for, in the pandemic, actually getting multiple people there doing the sampling. And this is all modifications of things that I did during my master's project with Dr. Draney. And then we also looked at soil dwelling fauna. We did this by hand sorting monoliths. So we would dig basically a square-foot hole, right, 30 by 30 by 30 centimeter, pull out that chunk of soil, and then all lounge around on a tarp and sort through that, pulling out all of the invertebrates that we could find. Okay. This is a project my graduate student, Madeline Olive, has been spearheading. She should be wrapping it up in the spring, and she's working on ideas as fast as she can. We have some preliminary data, and so no statistics today. Just going to show you some of the trends that we've seen, and I'll kind of walk you through this graph. And I got a couple of these graphs, but essentially we have our 3 sites. Rough Ridge is northwest Georgia, Rock Mountain Fire was northeast Georgia, and Chimney Tops 2 up in the Smokies. And here we just have total abundance of all taxa, unburned in green and burned in gray, across time for each of those. Okay. And for the most part, there's no obvious differences, although occasionally at some sites and some seasons, you do find there's probably a big difference there. That's very coarse data. Well, I guess this is total abundance, so this is accurate to what it's going to be. We have this for litter, and then we have this for soil. Again, a few places we start to see some differences, but this is sort of standard for ecological data. It's messy. Okay. And as we get deeper into it, we're going to see some more things, especially as we look at those particular dates and times. Almost more interesting to me is the data on diversity. So this is really coarse level diversity, so spider, ant, millipede, right? And we're right now working on refining those IDs. But even at this coarse diversity level, we're seeing some differences, and only at occasional times. But this indicates that there may be some things happening. Okay, so we see this in a few places for litter and also in a few places for those soil fauna samples as well. Okay, and sometimes we start to see them a lot. And so we may start, as we dig into this data more, especially as we get to IDing the millipedes species, I think we'll start to get some really interesting things out of this project. Okay, I think I have come to the end of my time. There's so many more projects that I wanted to talk about that I just knew would not fit in here. But I have a ton of people to thank. Right? Anytime I said I in the last hour, really I should have said we, because I didn't do any of these projects by myself. I've had tons of student involvement. I've had a lot of different funding, multiple sources of internal funding at Georgia College, which has been really fantastic, as well as institutional support and external funding from a lot of the institutions that you see pictured here. And with that, I will leave you with a couple more really good pictures of my favorite fauna, and I would be happy to take any questions.
People can unmute themselves to ask or chat questions in.
Should we raise hand, or I don't know how to do that.
Why don't you just go ahead and talk? I mean, if we hear two people talking, they can sort it out.
Hi, this is John Luchai. I'm a geoscientist. I enjoyed the talk. I had a couple questions. I had a call in the middle, so hopefully I didn't miss this. But you mentioned that the millipede diversity, I think there are 1,500 species in North America and 15,000 across the world. And I kind of, while you were talking, I looked up the areas of land on the world, and it looks like we've got something like 10% of the world's land area, or 10% of the millipedes, but we have like 18% of the world's land area if you ignore Antarctica and Greenland. So really we're very underrepresented, despite the fact that we probably have a really fairly healthy research endeavor, right? A lot of people can go and do this research in a university-rich environment like ours. So compared to other places, why do we have such a low relative diversity? Is it because half of North America was glaciated and is in the colder climates, or is there something else going on? That's one of the two questions.
Yeah, well, let's start with that one. It's a great question. So it's funny because this is kind of how I got started. So I went and did undergrad research at Toft Point, and we found a bunch of stuff in a bunch of different, there's lots of different habitats there. And then we looked at the species list for Wisconsin and said, where are all of these species? And so I got really, really fired up about biogeography. So I think the answer here is there's a couple of different things happening. One is that there are a lot more species in tropical regions, and we don't have a lot of tropical regions in North America, depending on where you cut off North America. But the other thing is, even though we've got a really great scientific infrastructure, being a developed country, we do not have a lot of people who work on millipedes, and we have not historically. So in terms of millipede ecology, so in terms of ecology, there are 2 labs. In terms of taxonomy, there's maybe 5 of us right now in North America. Globally, so I've been heavily involved in the International Society of Ecology, and there are about 170 of us. And so we do not have a lot of people doing this work. I think that's part of the challenge. 5 might be an underestimate because we just had a couple of PhD students graduate out of the Virginia Tech Lab. And so I'm hoping that we start to see a bit of an increase in that as we can get more people fired up about it. But I'm sure there's a lot out there really to describe.
Would I be able to ask a second follow-up?
Sure.
So, I know this is something that's difficult to do and time-consuming and so on, but since there's an effort by at least a small number of people to increase the knowledge of our species that are out there, are people keeping are people getting a DNA database? Are you doing the genome of the millipede? Are you trying to actually categorize their DNA as well as just the morphological species descriptions? Or is that not
Yeah.
really something people are doing yet?
There is a big push, as with other taxa, to really go in that direction. That's never been my background. It's always been a challenge for me to try and incorporate more of the molecular work into the ecology. But it's something that even we're doing. So with this fire project, I was able to get a little bit of money in that to do molecular work. And we're basically just trying to establish a set of barcodes right now. But yeah, with the people who are doing more of the taxonomic work, and there's a lot of people doing phylogenetics and fewer people doing alpha level descriptive taxonomy. So everybody that's doing phylogenetics is doing a lot of molecular work.
Okay, thanks.
Mike, I think you can go ahead and.
All right. Okay. Yeah, I really enjoyed it, Bruce. I was hoping you might be able to give me a little update on something that we used to talk about when you were a student. Obviously, the earthworms have a real obvious impact on the forests up here and probably on the millipedes. But when you were doing your work, we definitely speculated that terrestrial isopods might be important because they're sort of maybe ecologically similar to millipedes, and that they might be displacing. We had a huge list of historical records of millipedes from Wisconsin, and you were able to find very few of those species. So I'm wondering if anyone's working on terrestrial isopods as invasives, and if we know anything more about that than we did 10 years ago.
Yeah, that's a good question. I don't think we know much more about that. I did a little bit of isopod work when I was in Kansas, trying to look at what was the prairie, essentially. And I had a student work on a seed eating project to see if they would eat seeds.
Yeah.
But I think we're in even worse shape with the number of people that do terrestrial isopods. You know, there's plenty that do the marine or the freshwater ones, but
Yeah.
I don't know of anyone that does. And admittedly, I have not been as active in that part of it, but I don't know of anyone that's actively working on terrestrial isopods in North America.
I don't either, Bruce. It's really amazing, isn't it? It's kind of shocking.
Yeah, so our colleagues at the Milwaukee Public Museum have retired, which I expect that they have. Then, yeah, we're in very rough shape with that.
One retired and one died, Bruce. I'm sorry to say. I can talk to you about that later.
Yeah.
There is a written question from Bobby. I think I would from Bobby Webster. But do you see that, Bruce?
It's in the chat there.
No.
Do you have recommendations for forest management in Wisconsin regarding invasive earthworms?
Ooh, this is something that's big time on the list. I know when Brad and his colleagues found Amenthus in the UW Arboretum and around there, everybody got super concerned about it. The challenge is once the earthworms get in, you can't get rid of them. Right? There's just no way to exterminate them. There's no vermicides that aren't just broad spectrum kill all the invertebrates, and you don't want to do that. So it's mostly about prevention. Get people to either not use earthworms as bait or to be very responsible with killing any bait they don't use. Watching how earthworms get moved around. So all of the earth moving things, cleaning truck tires between sites, selling plants as bare root if possible. I know it's a lot more complicated, but a lot of places in North Georgia that we see these worms moving in are probably places where there have been horticultural plantings. We haven't confirmed that, but that's one of our thoughts about places where one way that they're able to spread so much. So if you can prevent that jump dispersal and stop the human part of it, then I think you've got a pretty good shot at slowing things down. Does that answer that question?
Yes, thank you.
And then Bruce, there are a couple other or comments in the chat window. One from Vicky Medland, great talk, Bruce, great to see you. And another from Bob Howe. He said, excellent presentation, Bruce. Keep up the good work. All the best to you and your family. I have a meeting at 4, so I have to sign out. So it's too bad
Thanks.
you couldn't stick around. Do people have other questions for Bruce?
Yeah, I've got a question. This is Kier Wefferling. Great talk, Bruce. That was awesome. I appreciated all the natural history. Actually reminded me of a talk that I saw Mike Draney give not too long ago. And I was just wondering. So first of all, earthworms are they're a clade, right? They're one lineage.
Yes.
Okay, cool. So I mean the diversity was amazing. The morphological diversity. And I guess I'm wondering what proportion of the earthworms in North America are native versus non-native, because growing up as a gardener, they were always the good guys, and then I started to see the destruction that they wreak in nature.
Yeah. So in terms of the earthworms,
Yeah.
Of the species that we know and have described, because there's a big backlog of undescribed stuff too. It's about 30% are non-native.
Okay.
And that's sort of continent, well, U.S. and Canada wide. Mexico has additional native species as well. Yeah, so it's pretty significant numbers. It's a bad ratio, put it that way, as far as earthworms go. With millipedes, there's only a handful of non-native species.
Okay. And are most of those non-native earthworms ecologically problematic, shall we say? Are they bad?
I'd say probably.
Okay.
But if I had to show you data for it, then it would be tougher to show, mostly just because we haven't looked at a lot of them. So we know the Amenthus species come in and change things. We know that for sure that there are probably 4 of the Europeans that can make major changes.
Okay.
And then the rest are probably.
Mm-hmm. Okay, thank you.
Yeah.
Other questions for Bob or for Bruce? Excuse me. And thank you, Bruce, for inducing our putting up with our curiosity here.
Oh, I can talk about this stuff all day.
Indulging was the word I was looking for.
Yeah. I'm always happy to answer these questions. So I've left my email up there, and I know at the beginning I had some of my social media stuff. So anybody's welcome to contact me with questions about these sorts of things.
Yeah, thanks for the talk. It was great.
Yeah, it was very good. Thank you. So thanks again, Bruce, and thanks again to everybody for turning up. And I hope everybody has a good weekend. Thanks again, Bruce. I appreciate it.
All right. Thanks, everyone. Stay safe out there.
Bye.
Thanks, Bruce.
Thank you.
I enjoyed it. Yeah, you still on the line, Bruce?
Yeah.
Oh, there he is.
Simplify things a little bit here.
Bruce, I don't think I can put on my my video anymore, but yeah, I don't know if you had heard about Joan Yass. That's who I was thinking of. She had cancer about a decade ago and died. So she's probably the main isopod worker, and I don't think anyone else is really doing it. No one's else really taking it up. So yeah.
Yeah.
Hey, I
wondered about them. I wondered about them, and I still use their all of their resources, really. Their atlas and checklists are that's what you have to go on. Until somebody sits down and does that, you're kind of on your own.
Yep, absolutely. Yeah, I got to get that time and hierarchy, biological hierarchy graph from you. That's a really cool graph.
Yeah,
Um,
can do.
I'm not sure if I have your paper actually, because it came out in that book, and I don't. I'll have to.
I can send it. I can send it to you.
Okay, awesome.
Or if you want the book,
Thanks.
I can send that to you too.
I'll take the paper to start with.
It's a smaller PDF.
Yeah.
We'll write that down
All right.
before I forget.
All right, great, thanks. And
Yeah,
that was
came
the same
out.
photo that I have. The photo that you showed of yourself is the same one that I have of you. So apparently it's the only evidence.
Yeah. I've been thinking about that for the last day. Like we just didn't take a lot of digital pictures anyway. And
Nope.
I didn't focus on taking pictures of people until much later. Like I always took
Yeah.
pictures of things. That's just always something I had done. But especially you're out in the field, you're not thinking about that stuff.
Super busy. And during your undergraduate, people didn't even have phones to take photos.
Yeah.
So,
Yeah. Well, at first that's what I thought it was, and then I realized that I found a folder that said pictures already burned, and I thought, oh my goodness, I must have this stuff on CD. So I had to find some of the old burned CDs of data back from '04.
Yeah, I've got stuff on CDs, and I'm not sure if I have a CD player anymore on my new laptop. So
I had to find one.
Things are gonna get lost that way. Yeah.
Yeah, but I found all that data. I found all the pictures from the field. So there's not much else in there that's good.
Yeah. Well, it was good seeing you, Bruce. Thanks again.
Yeah, likewise. Thanks for having me.
Yep.
Yeah. Hey, Bruce, I have one more piece of business to discuss.
Yeah.
We can give you an honorarium for your talk, and our standard payment is $100.
Cool.
But what do I want to call them? The savages over in the finance department make it hard for us. So I will need a signed hard copy of I forget what it is, a W-2 from you, I think, or something like that. Anyway, I'll send you a form, and then sadly you'll have to mail it up, and it'll move at the speed of a glacier. But eventually, then a check will be cut and mailed back to you. So I apologize for the extra hassles, but we do appreciate your effort.
That's pretty standard state institution kind of system. If it's got to be a paper check. So
I was hoping,
that's fantastic.
paper checks is the problem. I was hoping with the COVID stuff they would let us do an electronically signed W-2 or W-9. I always forget what the form is called, but no, everything still has to be on chopped dead trees. So chopped pressed dead. Okay, well, have a good weekend, and thanks again.
Yeah,
thanks, Brian.
Thanks for having me.
All right, take it easy.
All right, you too. Be safe out there.
Okay.